All right. We're continuing with how we can analyze RNA-seq reads. So generally, there are kind of two pathways we can choose to go down, and it's kind of dependent on if there's a reference or not. So if there is a reference, for example, we know all the CDSs that exist in a species, right? There's a reference on ensemble. There's a reference on NCBI. What we would do is just clean our reads up and then map them back to the reference and start counting. We'll eventually do that, but if we don't have a reference, which is probably still a little bit more common in comparative genomics, then what we have to do is assemble those reads. And so it's a little bit, the idea is reasonably simple, and it's basically to just find overlapping reads that share sequence similarity and more or less build a contig from that, right? Just a contiguous sequence of reads that overlap with one another and produce what would be a transcript. And so the whole point is we've done a transcriptome sequencing, and so we can use assemblers to either make a transcriptome assembly. And again, it's just basically reconstructing all of the sequences, all of the transcripts that are present in our tissue. And so that's what we're going to do first. Again, it's just because either way we're going to have to map reads back to our reference. But if we don't have a reference, we're just going to make it. So currently, I'm just going to assume that we don't have a reference. So we're going to make it. Even though this is human testis, normally, right, if I were doing this for real, I would just map it back to a reference and be done. But for all intents and purposes, we're going to construct an assembly. So I'm in my FASTQ folder, and I removed all my unpaired reads, right? I don't care about the unpaired reads. I'm not going to use them. I don't need them. I already have enough data from the paired in stuff that I can just work with all of my clean reads and be done. And so surprisingly, I think transcriptome assembly is a reasonably polite process. There are 12.5 different assemblers, but my favorite is still Trinity. So Trinity, I think, was published and produced, I think, in 2013. There's definitely more of them, but I think Trinity is my favorite because it's simple to use, the output is easy to interpret, and there are definitely downstream tools made by the Trinity team that makes analyzing that output pretty polite. So Trinity was produced by a team at the Broad Institute, or the Broad Institute. This is like a collection of nerds at MIT, Harvard, etc., to analyze sequencing data. All right, they're still there. And so this team made Trinity. And so I do already have a Trinity instance running. Unfortunately, I started it this morning, hoping it would finish before class started, but I ran into some problems with an input file. Like, one of my input files was a poor format. I had no idea how it happened. So I had to rerun Trimmatic, remake my clean reads, and that seemed to work. I have no idea why it came out wrong the first time, but nonetheless, if you kind of see the same thing, like a failure of Trinity because of the input reads, just re-clean them and try again. Hopefully it will go away. All right. So since that's running, today's probably going to be pretty short. All I'm going to do is just demonstrate how to run this line, and it does take a while. So far, just using 6 processors. That's all I'm going to use for this. It's going to take a little while. So definitely make sure you run this in a tmux shell. Otherwise, you run the risk of a computer network disconnecting before your job finishes. So it's best to run this in a tmux shell. And so Trinity is hanging out in opt as well, just with a few other programs. So if I list opt, right, the things that are already there, right, FastQC is there. I have a few other programs unrelated to this class up here, but Trimmatic is here, Trinity is here, and another program that we'll use called transdecoder is also here. So all things are in the opt that we're going to be messing with for the next few days. So Trinity is in the Trinity dir. No. And it's pretty easy to call. Right, the only program that we have to worry about in any of this is Trinity with a capital T. So make sure whenever you type out Trinity, you do start with a capital T. Otherwise, you won't be able to find it. And so Trinity kind of has like these three stepwise processes. It's kind of, and honestly, they play on the birth of a caterpillar. So you have an inchworm, a chrysalis, and a butterfly, right? So that's kind of the order of operations. We're going to do an inchworm's going to run first, chrysalis is going to run second, butterfly runs third. Not super important to understand the details, but I did provide a couple of videos that describe what's happening behind the scenes in Trinity. I understand what's happening, but it's hard to explain, especially without drawing anything out. It gets a little tricky to try to explain. But again, the whole point is to just assembly these reads into non-redundant transcripts, right? Unique transcripts that are present in this whole data set, just by finding overlapping reads that share sequence similarity, right? I mean, that's the gist of it. The computer wizardry gets way, way, way more complex. Nothing I can replicate. So I'm bound to using these tools. And so the help menu of Trinity is pretty polite. So if I just run, as I said, I have this running, so I'm not going to run it again, but it will set up the line to run. So I have to call Trinity from its home. And again, like, the help menu is going to be pretty polite. Really, like, all that you have to provide in a Trinity line is just that. That's it. That's the only line that really matters. However, there is an option I should be aware of. I just need to scroll up a little bit. So you Trimmatic, like if you have Trinity outlined and set up right, you can just simultaneously run Trimmatic first. So it will take your raw reads, and you can make Trinity run Trimmatic. I think it gets a little tricky, so I keep them separate. That's just my preference. Let's see. Yeah, so all the main stuff. SSLib type. There we go. Yeah. So this option SSLib type. So you have to expect that all transcripts should be in the same orientation. They should be in the 5-3 end, right? And so your RNA-seq stuff should also mirror that 5-3 end. And so we can kind of, whenever we do like a CDS or a cDNA reverse transcription, depending on how the library is made and how this stuff is isolated, you can kind of tell Trinity to look out for transcripts and to keep everything in the same orientation, keep everything 5-3. And you can do that with this SSLib type. And so specifically, you can either do, it's kind of like saying what's first. If these are paired in reads, are you looking at the reverse sequence first, and then the forward sequence first? Or sorry, reverse then forward, or forward then reverse. But if you do this DUPT, so what this DUPT means, well, I kind of forgot what that means. But most library constructions are DUPT. or DUTP. In which case you do like an SSLib type RF. I think that's more common than not. If I'm wrong, let me know. But I'm pretty sure SSLib type RF, because the first read will be in the reverse orientation, and the second read is going to be in the forward orientation. Anyways, so we'll just kind of look at the details of this line to make it work. But again, all that we're doing is this plus an SSLib type RF. So given our reads, Trinity, Trinity seek type, we have to tell Trinity if this is a FASTQ or FASTA file, and everything's FASTQ, so it's just FQ, right? Seek type FQ. Max memory is required by kind of an independent algorithm. It was called Jellyfish. Not super important, but assemblers require a lot of memory. And it's almost, you kind of estimate how much memory you need by the number of reads present. I kind of forget the relationship, but I think for every million reads present, you should assign 2 gigabytes of memory. So we have a decent amount of memory to play with. I think I'm running this currently in my other job. I'm keeping it at a max memory of 20 gigs. And I think that's a fair starting point. Trinity will let you know if it's out of memory, in which case you can redo it with more memory. But I think more often than not, max memory for transcriptome assembly should be fine using 20 gigs. So it's just 20 gigs. To say that we're reserving 20 gigs of memory out of whatever total we have. I think we have 128 to deal with. But so far, 20 works. I haven't tried anything less. I bet I could do it with 5 or 10, but I haven't tried it. Left, so this we call in our paired in reads. So left is going to be clean 1. Right is going to be clean 2. And CPU, just how many processors we want to use. Since we have multiple people using this server at once, recognizing other people are running jobs, other classmates are running jobs. Ultimately, this maxes us out at, if all of us are doing this at the same time, we get maxed out at 140 gigs and 42 processors, which is about maxing that machine out. So this is definitely something we should be aware of. Like, don't go above 6 processors because you don't know who else is running something. And then SSLib type. Gosh, I forgot how that's. I think it's lib type RF. And so that's how we would run this line. As I said, I'm not going to run it. I'm just gonna leave it there because I already have this stuff running out somewhere else. And so we can at least see what it looks like whenever things are going correctly. So I have this going in a different window. So I'm just going to scroll up a little bit. Let's see if I even have that problem to begin with. So yeah, I think this is my good run. Everything's going fine there. Yeah, I had to rerun my Trim-O-Matic because something stupid was happening. Something stupid was happening. And so I would get an error. So I think that this is the first time that I ran Trinity here where I got an error. So at least it kind of understanding the errors that you can get might help you with troubleshoot the problem. And so I got this stupid error. Yes, here. So I got this error here. And so one of the first things that it's doing is it's going to convert these FASTQ reads into an into FASTA, which basically is going to get rid of the quality scores and additional information that we really don't care about. These are clean, so we just assume they're good. And so right, it's going to run this program gunzip on clean FASTQ1, but also turn it into a FASTA file. Same thing for clean2, right? It's just going to turn it into a FASTA. However, I got this stupid error with a specific read that's saying that the quals in the seek didn't line up. Whatever. If you see that problem, I don't know why I got that error, but it seemed to be a Trimomatic problem. Trimomatic put my reads in a weird, wonky way, or I was doing something to this file while Trimomatic was running, and I got a problem. Either way, I got a corrupt file. It threw a hissy fit, tried to continue, and then just said I can't do this anymore and quit. So that's possible. One problem. I eventually found that error and redid my Trinity, or sorry, my Trimomatic, fixed everything up, and got rid of the unpaired and ran Trinity again. And that seemed to work everything. I didn't run it with the SS libtype. I may run it again with the SS libtype, so everything that you do is similar to what I do. We should mirror each other, right? But nonetheless. So the first one is clustering RNA reads, trying to get everything sorted. And we also find k-mers. And so in other assemblers, so I sent out a video that describes what a k-mer is. And it's just a sequence, a specific sequence of length k, and that k is interchangeable. In other assemblers, you can adjust your k. It can be anywhere between like 21 to 37. But Trinity has a k, just inherently sets a k of 25. That's just right. Inherently sets a k of 25. And so what that means, if we have a 100 base pair sequence, we're going to try to find all of the unique sequences of 25 bases in that one read. That's probably the only thing that I know how to do from this assembler, is to take reads and split them into unique k-mers. And so that's the first thing that this program does, is find all the unique k-mers, all the unique sequences that are 25 base pairs long throughout all of our RNA-seq data. Yeah, so that's kind of the first thing that happens. And that should finish, and then you should get a jellyfish thing. Again, it's going to catalog all these reads. And then the second step should be Chrysalis. And Chrysalis will eat up a lot of space. Recognize, so this whole program is going to occupy a lot of hard drive space that we'll eventually have to clean up when this is all done, because a lot of it's going to be garbage that we'll never look at again when this is all said and done. Yeah, so right, so Jellyfish building a k-mer catalog from our reads. The specific k-mer is 25. That's not adjustable. That's just set by Trinity. But it also makes our life a little bit easier because we don't have to try to mess around with our k's to get an optimal assembly. We're just going to get what we get. And it's usually right. It's pretty accurate. Okay, then Chrysalis. So Chrysalis is actually going to construct the De Bruin graphs from all of these k-mers. And that's a little bit harder to explain, but this is the step where we're trying to find overlapping reads. But things get very weird in constructing a De Bruin graph. Again, I have trouble explaining it. I certainly have trouble explaining it without drawing anything out. But those videos should kind of help, conceptually help you understand what's happening whenever we're constructing our De Bruin graphs. But we're constructing a De Bruin graph for each transcript. Which is, again, it's just going to give us a complete assembly of a specific transcript. So when all said and done, we'll have like 100,000 De Bruin graphs that we will construct and have to search through, because we'll have about 100,000 different transcripts available. So Chrysalis, and I'm still in Chrysalis. So that's where I'm going to be for a while. I'm just going to let this run. So on Wednesday, we'll look at the output of this file. So again, if you want to construct your transcriptome assembly while we're waiting, feel free to, because once we have all those transcripts ready, we'll move on to the next steps of analysis, which is looking at how many reads, how many transcripts are present, where the open reading frames are, like identifying the open reading frames, and annotating, right? So even whenever all said and done, all we're going to see is the transcripts. We're going to see 5 and 3' UTRs in HEG to stop codons, but we'll have no idea what that gene is. Right, so that's going to be the next step, is Trinity to assemble, find the open reading frames, and then annotate those open reading frames to a specific gene. So that'll be the next step. As I said, this video is going to be a little bit short, just because this stuff is still running. But yeah. And so, as I said, this stuff will eat up a lot of space, right? So I started with, if we think about my starting material, there are only 2 gigabytes worth of data and all of that. But by this point, this in silico renormalization folder is 33 gigs, right? I started with 2 gigs of data, and my output so far is 54 gigs, right? That's too much space. We're going to run out of space pretty quickly if we leave all of this junk here indefinitely. So whenever your stuff is run, we will have to get rid of this garbage. A lot of this garbage. There's really only one file that we'll care about when this is all said and done. All of this other stuff is just supplemental that nobody ever looks at. I certainly have never looked at the output. Except for the one file that this makes that we need. But yeah, I'm probably going to leave it at that. I guess we can look at the K-mers. So we can look at this jellyfish K-mers. Let's see if, I'll just head that file real quick. I'm not going to look at it. I'll just head that real quick. Yeah. And so, yeah, nice. These are all the unique K-mers, and this is also, it counts how many times that K-mer is present. Let's just get 20. Let's get 30 lines of this. Okay, and so each of these reads. So we start out with like the very first, most common read as a poly A tail. No surprise there. We have we poly A tailed selected our mRNAs, right? And so this K-mer was present about 11,000 times. Probably a little longer than that, but 11,000 times. This specific mer was present 1 time. That was there 2 times. And so we will we have to care about how many times a mer was present. And that is something that jellyfish and Trinity as a whole will deal with, right? So this is a pretty common mer, present 127 times among our RNA-seq reads. And so this information does ultimately come out. But again, we basically find, jellyfish is going to find all of the unique mers, 25 bases long throughout our entire transcriptome data, right? It's going to sort through every single read, identify the unique mers. To give us that file. And so that we don't really we will eventually use the RNA-seq data later, but when we're building the assembly, when we're building the De Bruijn graphs, we only care about the mers, right? All of these unique mers. Yeah, I don't think we need anything else. Let's see what the histo is. Yeah. Oh, oof. Yeah, I'm not going to deal with that. Not going to deal with that. I'm going to leave it at that before I embarrass myself. So we'll pick up with this more on how to parse this output, what we actually produce when all is said and done, and how we can analyze it further. So we'll call it at that for now, and I'll see you all again on Wednesday. Feel free to let this line run until it's finished. Just do it in a tmux, because a lot of the processes that we're going to do now are going to take hours, right? They'll just take hours to run. So you can kind of set it and forget it, run it in a TMAC shell. Anyways, I'll leave it at that and see y'all again on Wednesday.